All right, everybody, let's go ahead and get started. Today is September 29th, and this is Biology 105. Thank you all for making it here once again. It's so good to see you all. I hope you had a great weekend. I hope the weather's treating you all right. It's great. We all love it. We all love it. Okay, so here we are in the schedule. It's Monday, so let's go ahead and orient ourselves to the course. You're in week 6. You know, you're getting close to that sweet fall break. It's coming up in week 12, so we're halfway there, right? Which is pretty cool. Wait, no, we're not. Never mind. It's like in week 14, so it's forever from now. But we're getting close. Yeah.
Isn't the end of like this semester like 15, 16?
Yeah.
Why is it? Okay.
Hey, don't ask me. I think it's wild. It's the craziest thing about fall semester is you've got this block of time from week like 6 to week 12 where there's just like no end in sight, and it can be discouraging. But we're all in it together, and how's the song go? Wildcats everywhere, throw your hands up in the air. Okay, cool, we're having lots of fun. Good. So here we are in week 6. We're on a vacation for a little while, but we'll get through it. We get through every fall semester, bless you. You don't have an exam until week 10, so you've got a little bit of time where you're just going to be focusing on coming to lecture, going to your dang labs, doing your lab assignment, doing your reading quizzes. Just keep it up. We're going to be covering plasma membrane stuff from week 5 today and probably a little bit tomorrow. And hopefully we'll be caught up with material by the time we reach exam 2. We always are. That's how it always goes. So you just keep up with your reading as assigned, keep up with your reading quizzes, keep making it to lab. This week's a pretty cool lab where you're going to be doing gram staining. It's an essential technique in, microscopy and characterization of small organisms, single-celled organisms. So I hope you really enjoy this week's lab. It's cool. All right. Good deal. Then we do potato lab. It's a classic. Photosynthesis lab. It's a classic. Strawberries. They're a classic. It's all classics from here until exam 2. Any questions now that we've sort of just, I'm just telling you, like, hey, orient yourself to the course. Yeah? You have a lab midterm coming up. Thank you for bringing that up. I'm so sorry I forgot to mention it. Here it is, lab midterm. Where are you? Here we go. In week 8, okay? It is an online lab midterm. It is open notebook, open notes, open internet, open everything, okay? And you got, I believe I have 2 hours for you to take it. Most people complete it in like 30 minutes, okay? The idea is I want you to be able to see what the lab final is going to look like, because in the lab final, which is in our last week of lab, it is open lab notebook, but that's it. And you have to do it in lab during your regular lab period. So this online lab midterm is going to serve to show you sort of the types of questions and the topics that you're likely to see. And it will also, but it will be all multiple choice, drop down windows, fill in the blank with a number sort of stuff. So be prepared to do shorter answers on your lab final, which I'll bring up more and more. But everybody's keeping a lab notebook, right? So you're all doing great. Yeah, everybody's got a good lab notebook with lots of notes. Did that answer your question? Awesome. So there it is. It's online. We can even look at the assignment to make sure everybody's cozy with it. Yes?
Is that due during lab or like 5 minutes?
I actually forgot what I set the availability for. So let's take a look. It should be in week 8, online lab midterm. Assignment name, online lab midterm. It's just as an assignment. Where's my quizzes? Where's my quiz? It's supposed to be available from October 13th until October 17th. But I don't have, oh my gosh, this is a really good thing that we checked this, because I have the quiz, but this is just the assignment, which is different. Canvas is so great, from the actual quiz, which is probably under my quizzes. I'll sort this out right after class today. Lab midterm. Oh, whoa, what if I do this? Is this helpful? Does everybody see it? This is, I think, just the assignment, but this is just the assignment. Everything's so difficult. Let's see. I'll find it. I have an assignment made and built. That's not it, though. Yeah, that's when it should be due. It should be available for you during that week. Yeah. But that's not the official assignment, so I have to like find the actual assignment, which is apparently like not published. It's going to be a headache, but I'll sort it out. That's my problem. Anyways, 13th through 17th. All right, solid? Cool. I'll post announcements as we get closer to it too. Yes?
So for the first exam/quiz we did in class, you said that you would have availability to meet with us.
Yeah.
And discuss it. I'm assuming that the midterm isn't, because I know that some little quizzes on Canvas you can go back and see the answers and stuff and see what you got wrong. Will that be available?
It'll be available. Yeah. This isn't meant entirely as a study tool for you to prepare for the lab final, really. It's less of a midterm and more of a, every time I do this, most everybody gets 100%. Because even if you don't get it, you can Google the questions and put in the answers and get 100%. Well, I'm just telling you that I'm spitting facts, all right? It's worth 50 points. The lab final is worth 100 points, and plenty of people on that lab final get 12% because they didn't actually use this as a review thing. They looked at it as an assignment and just did it with what I'm telling you right now. So 100%, you can do that. You can 100% this assignment just by Googling everything. But if you don't understand the material at all, you'll probably fail the lab final.
Will you be meeting with people to discuss?
Absolutely. That's what I got office hours for. You come. I'm so lonely during my office hours, everybody. None of you come and visit me. You just leave me alone to wallow. Thank you so much. And I'm already up to season 5 of shows that I'm rewatching during then. So, just come by. It'll be fun. And I'll cover anything. I'll open up lab midterms. I'll open up exams. I'll open up assignments. We'll look at how you did. Totally. Solid?
Yeah.
Great. Yes, please.
2 tries quizzes or is it 1?
One try. So it's the one attempt thing because it's open everything, right? You get one shot at it. And yeah, the average, the scores usually range from 40 to 50 out of 50. Yeah. Question. Sorry, I'm running. Yeah. Well. Yes, the lab final covers everything. And if you have a good lab notebook and you've been keeping lots of good notes, I get plenty of people. The distribution, it's another bimodal distribution, right? Where there's people here who got 40 out of 100, and there's not very many people who get between 40 and 80, and there's a ton of people who get between 80 and 100. Every semester it goes that way. That's just how it goes. Yeah, there's very few people who get C's on that. You either fail it or you get a B or above. Yeah, and it's all based on preparedness and how aware you are that it's coming, So everybody's aware about that lab midterm and about that lab final mainly. It's 100 points. It's a good amount of points. Cool? Awesome. Question?
Yeah, how many questions-wise do you think the lab final will probably have?
Last time that I did the lab final, it was, I think, 10 pages, but some of those pages have a single question. So I think it had 20 questions on it, where 8 of those were multiple choice. But then some of the questions are here's the description of a population's genetics. Boom, boom, boom. Here's how many homozygous dominant, heterozygous, homozygous recessive individuals. What are the phenotypic ratios? What are the genotypic ratios? Is the population in Hardy-Weinberg equilibrium? That's 2 pages. And it can take 10 minutes or it can take an hour, depending on how much practice and prep you've done. But I'm not trying to blindside you on anything. I will fully explain every single topic covered that's going to be covered on the lab final. I even have a lab final practice quiz as your last assignment for the semester, okay? Which shows you more of those topics. And some of the questions are going to be nearly identical to those in the lab final. So the lab final can be a scary thing, but it's all about, are you prepping for it? Do you have a good lab notebook? You can put anything in there you want. You could print out an entire Wikipedia article. You could print out an entire, take screenshots of a tutorial of how to do this sort of math and print it out and put it in your lab notebook. All right? It's just all about how much you prep. Cool? Any additional questions about that stuff? Solid, folks. And also, like I said before, you got questions about any of this, you're nervous about any upcoming assignment, you come talk to me during office hours or make an appointment outside of office hours. I'll go through what's going to be on exam 2 with you. I'll help you fill out your study guide. My office hours are time that I get paid to work with students directly. You are the students, so don't feel bad about dropping by. I'm making money, okay, for that time, and I'm happy to do my job. I love my job, and part of it is working with you on an individual basis. Or in groups. Come in a group. If my energy's a little intense, which I've been told I can be kind of intense, you know, come in a group. All right. We're having fun. I'm having too much fun already. It's too much fun for a Monday, yes.
What's your opinion on camping, since it's also intense?
I love it. I love the joke. I love the fun that we're having right now, discussing the joke. For those of you watching the recording, it was a question about camping because you do it intense. Thank you very much. You can have a John point. All right. How are we feel? You ready to talk about what are we doing this week? We're talking about membranes. You ready to talk about membranes? Yeah. All right. Yeah. Here we go. This is going to be a ton of fun. So, here we are in the membrane PowerPoint. It's very exciting. The foundation of the membrane is going to be that phospholipid bilayer that we've been talking about. I got to actually double check and make sure that we did wrap our previous PowerPoint, because this feels new and I'm a little bit concerned.
I think we have more on cytostomes.
Do we really?
I left on centromes and
Damn it, the centrosomes. Okay, sorry, I got too stoked. We got to finish okay, only a few slides. Here we go. We did this one, yep. It walks by. It recommends all of the YouTube videos that make us feel concerned. All right, cool. So we finished talking about microtubules. Microtubules are hollow. Microtubules are really good for structural support. Microtubules are going to be incredibly important when we talk about cell division, because they are used to move chromosomes around. We'll talk about it quite a bit. But then again, during cell division, all of these cytoskeletal elements are going to be important because they provide structure to the cell. So when you're taking a cell, right, and you're splitting it into two, you're facilitating a ton of that with the cytoskeleton, okay? Including like a little band in animal cells that sort of forms in the center and pinches off the two daughter cells, we call them. And that's actually an example of a microfilament that does that. So while the microtubules are good for structural support, they resist compression really well, all right? They're involved in moving chromosomes around. The microfilaments and the intermediate filaments are very good as tension-bearing elements because they are like cables, okay? Multiple long strands bound around one another, just like a rope, really good at handling tension, okay? You ever played this game when you were a kid? I was in a very small town before smartphones and we didn't have internet, and so, you know, we had to make our own fun. What you would do is you would get a 12-pack of soda pop and you would drink all the soda pop and feel really sick, and you'd play this game where you would put the empty can on the ground, and you tried to balance on it without crushing it. So yeah, it's a fun game. Hollow structures are really good at bearing compression. And then a friend comes by and they flick it and move their hand away before their fingers get crushed. And that's the game, all right? Now, if I was going to try to pick up this chair, all right, I wouldn't the first thing I would grab would not be a hollow tube of aluminum that I would like adhere to this and then pull up, because that's tension. I would grab a rope and then pull up with that. It all comes down to the physics of how you bear tension, because forces get redistributed onto the adjacent threads. Is that stuff for your exam? No, that's just interesting tidbits. So microfilaments, they're the smallest ones. They're not hollow. They're like filaments. Intermediate filaments, okay? They're like filaments with more strands. They are intermediate in their diameter. They behave really similarly to the microfilaments. Okay? Microfilaments are made of tubulin. I'm sorry, not tubulin, actin. Microtubules are made of tubulin. Microfilaments are made of actin. These are all proteins, okay? And then intermediate filaments are made up of keratin. Keratin is something you've probably heard about. It's in your hair. And your hair is a pretty good tension-bearing element because of its, structure at the micro level of being made up of intermediate filaments. Okay, microfilaments. Muscle fibers. Muscles are made of proteins. We know this. Basically, you've got these strands of microfilaments attached to all of these myosins, these motor proteins, okay? So remember the motor protein, how it would get zapped with energy and it would like walk along a microtubule, right? Well, so now imagine that my feet, all right, and my hands, they're both attached to two different cables, okay? And then you zap me with energy and I go, oh, like that. That's a muscle contraction, okay? There's a cable at my feet, there's a cable in my hands. I pull both of them together with an expenditure of energy, and that contracts the fibers together. So microfilaments, okay, in an interaction with motor proteins, myosins specifically, okay, allow contraction of muscles. Please, question.
How would a cramp necessarily function in this? Like, because if it's like supposed to fire when you want it to go off, how would a cramp necessarily?
I honestly don't know enough about that. I know that cramps are, unintentional muscle firing, right? It's not like your autonomic nervous system isn't regulating that like contraction, right? But as far as what causes that, I'm just not knowledgeable enough to answer. Yeah. I'm so sorry, and I failed you. It was a great question. What are you even talking about? I just didn't know the answer. That's okay. Yeah. You know what? You're okay. I'm okay. We're okay. We're all sitting in here, and we're all okay. Okay. Here we go. Cytoplasmic streaming. So check this out. Actin filaments, microfilaments are really important in this process of cytoplasmic streaming, which is essentially, right? You ever, when you're in the pool, okay? You're splashing around, you're hanging out in the swimming pool. It's all so exciting. And you take your hand under the water and you go like, and you move a bunch of water around by moving your hand through, and then the current goes around you. Maybe you like spin in a circle and try to get the cyclone going, and you never can because you're just a kid. You don't know what you're doing. You don't know about, the physics here. You're just not strong enough. But someday, all right? Here's the thing. This is very intuitive and obvious. You move something through fluid, you move the fluid. I know I'm blowing your freaking mind there. Did the example help of being in the pool? I don't think it was necessary. But check this out, okay? You take all these microfilaments, you attach these motor proteins to them, you put big organelles on top, and then you just move those big organelles throughout the fluid. And what you're gonna do is swirl the fluid around, okay? So that's cytoplasmic streaming. Here it is actually occurring, okay, with all of these different chloroplasts being moved around. They're all attached to motor proteins, all attached to actin microfilaments, all right? And that's gonna churn the cytoplasm as well. That's beneficial. In this case, it's beneficial for a couple reasons, right? You make sure that all of the chloroplasts get light exposure by moving them around. You're moving them from the top to the bottom of the cell, for example. And you're also distributing resources and waste products throughout the cell, preventing them from accumulating in a certain region. And the same sort of principle applies to when you're moving mass amounts of cytoplasm throughout the cell to, say, extend a pseudopod as an amoeba. Pseudofoot, pod means foot. It's all very cool, but I would like to focus on the cytoplasmic streaming piece, which I think is more relevant for BIOL 105 as opposed to the single-celled organism question of cytoplasmic streaming for movement, for motility. Yes, please.
So, like, it's kind of like if you had like a pot of soup and you just stirring it around.
100%.
Yeah. So just because of like a salt moving through it, will displace like fluid.
Yeah, yeah. For something that's got such a fancy name, it's pretty dang, straightforward. It's like, you got a pot of soup. You want everything to be distributed evenly throughout it, and so you just keep stirring it, And so here you've got like these mitochondria are consuming, the breakdown products of glycolysis, for example. So you keep moving them so that they're constantly getting fresh cytoplasm surrounding them, and their waste products don't accumulate around them. Yes? The solid question. The nucleus is usually anchored in place by the cytoskeleton. Now, I don't have a great answer for which cytoskeletal elements are most important in nucleus anchoring, but there's probably an answer. And once again, I'm entering my admitting when I don't know something era, which is a big change for me, Yeah, but it's fun to take your understanding to its extent. And then be like, cool, now I have something more I could learn someday. I'm not going to do it. Maybe you could learn it. I'm done learning. All right? I gave up. No. Okay, cool. So let's talk about, we've been talking all about the cell, okay? Let's talk about the space between the cells, all right? So this is where we officially discuss the cell wall. We also basically are just discussing how cells are anchored to one another, okay? So with plants, it's a little more straightforward. They've got, plasma membrane, boundary between the cells, interior and exterior environment. Solid. Most cells put stuff outside of their plasma membrane, and this will either give the cell additional structure or let it be anchored to additional cells. Because once you, for example, have a mature skin cell, all right, on your body, I don't want, when I do that, for it to just go like bloop and like move around. I want it to anchor, okay, to adjacent cells. So in order to do that, you got to secrete some stuff outside of the cell. Let's talk plants first. I've told you about plants. I've told you about plants and their cell walls, how they build the cell wall outside of their plasma membrane. It gives them additional structure and stability, right? It lets them utilize turgor pressure to maintain structure in the absence of a skeleton, but it also locks you in place, okay? So the cell wall, all right, sort of locks you in place. It's made of cellulose, okay? And there's this region between adjacent cell walls called the middle lamella, which basically just fills up with glue, okay? It sticks the adjacent cells together. Primarily, this is a compound called pectin. Pectin is something that you may have encountered if you like making jellies, okay? Or even if you've shopped in a store for jelly-making supplies. People don't usually use gelatin anymore. Now, because it's made of animals, okay? We usually use pectin, which is a plant compound used for adhering things together. It's thick, it's gooey, it does the same thing, roughly. I'm sure there are people out there who are like, no, if you want a real jelly, you got to boil the hooves yourself, Someone feels that way, but I'm fine with the pectin. I'm fine with the pectin. Okay, so cell wall, solid, it exists. Plasmodesmata, you should be aware of. Plasmodesmata provide continuity between adjacent cells. They are essentially little hallways between the cell. And look, the plasma membrane is even kind of continuous between these, all right? They're tightly regulated. Not everything can freely pass through the plasmodesmata, and plasmodesmatal regulation is its own field of study. So what can cross, what can't cross, its own specialization. Here's a little actual image of some plasmodesmata, and check it out, right? The cytoplasm ends up being continuous through the plasmodesmata. So this has led some people to say that the plant is more like a single-celled organism that's been compartmentalized rather than a multicellular organism. But at the end of the day, that's really a debate of semantics. It's more nuanced, and so it doesn't perfectly maybe fall into either category, and maybe those people should just chill out a little bit, all right? It's clearly not a single-celled organism, but that's me, just coming down on them. How about we all just have our own ways of describing this? That's okay. Let's just talk about what's actually going on. Continuation of cytoplasm between adjacent cells, because you can't move things across the cell wall. Does that sort of make sense? It's easier to diffuse things when you're an animal, okay? Diffusing things from cell to cell is a little bit easier because you don't have that big polysaccharide, that thick polysaccharide coating around all of your cells, okay? But you also have to have a dang skeleton. So is it really worth it? How happy would you be if you didn't have to have this pesky skeleton? It's obnoxious. All right, questions. Animal cells, extracellular matrix. Animals do the same thing. They put a bunch of stuff outside of their cells. It's a lot of like collagen. Okay, you can see this collagen. It's another protein. It's a structural protein. So you put this outside of your cell, and then you sort of anchor your phospholipid bilayer, anchor your cell membrane to it, and that helps to hold everything in place, hold everything together, right? You know the areas of your body that have a lot of collagen in them, okay? All right. Very good, very good. You don't need to know these specific classes. The whole idea is just, look, you've got these protein cables, these protein structural components, and then you just anchor your cell membrane to it. Additionally, adjacent cells can be anchored more directly to one another, okay, through things like a desmosome or a gap junction, which is basically a plasmodesmata, but it's just made up of a protein. It's like a little hole, okay? You don't have to memorize any of these categories, any of these specific types of junctions. That is not the purpose of this. What I'm trying to tell you is, while we discussed the cells on the individual levels, right? We discussed individual cells. The cell does not exist in a vacuum. The cell exists by interacting with all of these other cells in order to make up a multicellular organism. The cells are anchored to one another regularly. Full independent cell motility is kind of reserved for blood cells, immune cells, things like that. Reproductive cells, okay? Solid. So this isn't about memorizing all the different types. It's not a cell biology course, even though we talk a lot about the cells, okay? But it's important to know that the cells are anchored to one another in different ways. How's that feel? Satisfying enough? Sick. Okay, we're going to talk a lot about this. So the cell membrane is a phospholipid bilayer with a bunch of stuff embedded in it, okay? And by embedding a bunch of these different proteins, you can give cells specific functions. So if all the cells have the same phospholipid bilayer structure, what makes them different? One answer to that question would be the proteins embedded in the phospholipid bilayer, okay? That's what's going to let cells, say, detect and respond to extracellular signals, or anchor to adjacent cells, or pump things in or out of the cell, all right? All this different stuff. Cool? So, cell membrane, that's what we're gonna, this perfectly transitions into a discussion of the cell membrane. That was great. That was great. I stuck the landing on that one. All right? I can't wait to talk about it. There we go. We got through all the slides. Let's just do a smooth move right here into membrane fluidity. All righty. Cool. Plasma membrane. We're going to talk about how a membrane's fluidity is maintained, because you don't want a membrane to be too fluid, it can fall apart. You don't want a membrane to be too rigid, it can shatter. So how do you maintain membrane fluidity? It's all about the degree of saturation of the fatty acids in the phospholipids making up your phospholipid bilayer. All things that we've talked about quite a bit, okay? Also, you can jam some dang cholesterol in there. and that'll help to stabilize your membranes. Components, selective permeability of cell membranes. It's really all about that hydrophobic domain. I have been telling you since the beginning, that's one of the most important principles here, okay, and you'll see why. Passive facilitated active transport, it's gonna be great. Tonicity, you're excited about it. I can already tell. You already had a lab all about it. Diffusion, can't wait to talk about it. Electrochemical gradients, super important. Photosynthesis, respiration, boom, here we go. What makes a membrane fluid? Well, a membrane is basically a bubble of oil, okay? But because of that special phospholipid bilayer structure, it's a stabilized oil bubble, where the interior can be aqueous and the exterior can be an aqueous solution, and these hydrophilic heads are happy to associate with those solutions. You get a nice hydrophobic region in the center that prevents the fluid from freely crossing, prevents things that were soluble in the extracellular solution or in the cytoplasm from just bouncing into or out of the cell, okay? The membrane's fluidity is maintained, or its structure is maintained because of those hydrophobic interactions, the hydrophobic interactions between the fatty acids of the phospholipids. So if it's a big here's one of my famous drawings of a phospholipid bilayer. Someday I'll get even faster with this, I hope. So remember, the little squiggly legs are fatty acids. They're made up of hydrogens and carbons, okay? All of this is carbon-hydrogen bonds, so it's all hydrophobic. There's no polarity to those bonds. Solid so far? Okay, like dissolves like, and this is behaving like a fluid, okay? Hydrophobic things can just dissolve into here, bloop, and then move around, all right? Because they're just floating in oil, and if they try to come out, they'll go like, oh, no, 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 and they'll go right back in there, all right? They don't want to go out because they won't interact very well with all the water molecules making up the extracellular solution or the intracellular solution. The cytoplasm, I should say. All right? Now, some molecules have a hydrophobic and a hydrophilic domain. All right? So check this marker out. You can all see this marker, right? Look, it's got a hydrophobic region and it has a hydrophilic region. I'll show you with pictures on the slides too. No need to worry too much. But check this out, right? This goes whoop and is soluble inside the hydrophobic region. Its hydrophilic region is happy to extend into the aqueous solution. So that's another sort of like an amphipathic molecule. It has a hydrophobic and a hydrophilic region. But it could flip like this. It could go, oh, I accidentally fell into the hydrophobic region. Oops, oh no, what happened? Boop. You see? So you can flip-flop. This is the point here, is that this is all very fluid, okay? It's not a super stable situation in the phospholipid bilayer. Things move around, okay? So what sort of things are embedded in the phospholipid bilayer? Well, proteins, cholesterol, glycoproteins, glycolipids. All right? What does that mean? So we've got the phospholipid bilayer structure. And I really want to just check in real quick. How are we feeling about that phospholipid bilayer? How are we feeling about this? There's a phospholipid. It associates and makes this bilayer structure. It's probably one of the most important molecular association things that we learn about, you know? The only thing that gives your body compartmentalization and order and structure is this, really. Yeah?
Sorry, can you go back a slide real quick?
You're darn right. It's just as easy as pressing this button. All right, solid. So we're all cool with the phospholipid bilayer? Okay, then I'm gonna stop getting so stressed out about it because you all understand it. So there's all this stuff embedded into the phospholipid bilayer, right? Including proteins, cholesterol, glycoproteins, glycolipids. It's just a sugar plus a protein in one molecule is a glycoprotein. A glycolipid is a sugar plus a fat, okay? And usually these molecules exist to do this. Bless you. Check it out. This glycoprotein, the protein is embedded in the phospholipid bilayer, and the carbohydrate, the glyco part, extends out into the extracellular solution, okay? You'll see in this demonstration slide that, yeah, there's nothing really extending in. And while that's not 100% accurate, it is good for our illustration here. Here's a glycolipid. Look, it's a fatty acid chain attached to a carbohydrate, ring structure carbohydrate, solid. These are super important as labels. Your body's really good at detecting carbohydrate labels and reading them. So cells will have these little carbohydrate labels sticking off of them, and then your immune system can read that and be like, oh, that's good, that's a friend. That carbohydrate label means friend, okay? And if it has a carbohydrate label it doesn't recognize on it, then it can detect that and go like, oh, I don't know what that is, let's destroy it. Let's tear it apart, rip it to pieces. I don't like things that I don't understand, destroy it, all right? That's what your immune system is like, not mine. My immune system is much more accepting. Okay, sorry, it's just a goof. So check it out, right? We've got the glycoproteins, the glycolipids. Primary function, self, non-self identification, or just to provide some labels. All right, it labels things. Cool? I love it. Okay, these proteins, that's going to be a big one. We're going to be talking about the proteins embedded in the phospholipid bilayer a lot in this lecture because they serve a lot of different functions, okay? Some of them are enzymes, all right? Some of them are like channels, okay, that things can cross through. Some of them are used to anchor to the extracellular matrix. They do all sorts of things. It's going to be a major focus of our PowerPoint. So don't worry about that right now. What you need to know is the terminology of a peripheral protein and an integral protein, okay? And then also a transmembrane protein, okay? So check this out. You ready? It's not so bad, I promise. A peripheral protein is bound to the periphery of the phospholipid bilayer. It's attached to the outside or the inside. A peripheral protein on the periphery. An integral protein penetrates into the hydrophobic core of the phospholipid bilayer. And that means that this portion of the integral protein that is embedded in the hydrophobic region must be comprised of mainly hydrophobic amino acids. Because remember that about our polypeptide structure? There's all those side chains. They can be hydrophobic. They can be hydrophilic. They can have a charge. They can be acidic and basic, right? They can do all sorts of stuff. Cool? Yes, please. One more time? Oh, I forget which ones there are, but there's a whole, their names aren't grouped in any way, but there's, some of the amino acids have hydrophobic side chains. So if you look it up, it'll tell you exactly which ones. Yeah, but I don't remember, I don't have them memorized. Yeah?
So just to clarify, integral protein is integrated into the membrane, but it has to be hydrophobic.
Exactly. That's great. So this region of the membrane is hydrophobic, so anything that is stuck in that region of the membrane must also be hydrophobic. That's key. That's really key here. Now, this is an integral protein, but it is also a transmembrane protein, meaning it fully spans the phospholipid bilayer. That's really important because if you want to, say, move something through this protein or transmit a signal through this protein, it needs to go all the way across the phospholipid bilayer and be a transmembrane protein. And if it's a transmembrane protein, once again, this region must be hydrophobic, and these two regions, here and here, must be hydrophilic. Otherwise, the whole protein would just fall into the hydrophobic region. How are we feeling? Question?
Is
I would say that a transmembrane protein is a type of integral protein. Yeah, there can be integral proteins that are not transmembrane proteins, like this one. This is an integral protein, but it is not a transmembrane protein. This is a transmembrane protein, but because it is also integrated with the hydrophobic region, it is still an integral protein.
So what would be the point of integrating with the membrane if not across?
Yeah. Oh yeah, bless you. Yeah, absolutely. So usually for like anchoring, or if you're an enzyme that say is, I got a good one. So if you are an enzyme and the reaction that you are facilitating, I'm going to be lazy and just say facilitating, catalyzing, that's the word I was looking for, okay, is an interior one, then you don't really want to be facing the outside, right? But you may also want to be more securely anchored than a peripheral protein, all right? So our important thing to know today is the categories. And this is another thing where, like, there's a ton of crossover between functions of all of these different categories of proteins. It is easier to come up with reasons that you have to be a transmembrane protein, because this can't pass things across. It's harder to come up with reasons that you'd want to be integrated, but not transmembrane. All right, yeah. Anchoring proteins, can be transmembrane or just integrated. So I don't have a very satisfying answer for you, actually. Really? Oh, awesome. I'm very happy to hear that. Okay, cool. That's the best answer I got for you. And if it works for you, I'm stoked. Okay, cool. Talk about this. We're going to talk about cholesterol's function in the phospholipid bilayer. It's really cool, but I got a slide about it to illustrate it. Remember, though, what's cholesterol? Oh my gosh, does anybody actually remember? That'd be crazy. Yeah, please. It's a steroid hormone, right? And so what class of biological macromolecules? Lipids, points. I'm throwing so many points at you, that's awesome. So just to repeat that back, cholesterol is a lipid, it's a steroid hormone, right? Well, it's a steroid hormone, and steroid hormones are all lipids, so they're all hydrophobic, so they're happy to dissolve into a phospholipid bilayer. That's going to be key when we talk about how those hormones function. It's also key for one of the roles that cholesterol plays in membrane stability. All right? Very good. Here it is. You're experts. I'm skipping past it. Okay, cool. So the phospholipid bilayer, right? Phospholipid bilayers, like anything else, you get them warm enough, they become a liquid. You get them cold enough, they become a solid, right? You get them really warm, they become a gas. You do not want your phospholipid bilayers to become a gas, okay? That kind of destroys the whole purpose. So cooler temperatures, membranes go from fluid to solid. You want your membranes to be fluid, okay? If they solidify, they can crack, they can burst. It's like all of your cells become a frozen bubble, and it's just no good. It's just no good. Yes, please.
What type of temperatures are we talking for freeze?
It totally depends on the organism you're looking at and the degree of saturation of the fatty acid chains. All right? So there's actually a massive amount of variability in the temperatures at which membranes freeze. But once your membranes freeze, life is really bad. All right? You really don't want that to happen. Cool. Good, yeah, so it all depends on the types of lipids. What I mean here is the types of fatty acids in the phospholipids. Okay? If you have a bunch of unsaturated fatty acids, specifically the cis unsaturated fatty acids, which remember, make the bends, okay? That's gonna help maintain fluidity. Do you remember when we talked about saturated versus unsaturated fats, right? Remember how a saturated fat is like butter, okay? A saturated fat is like a bunch of dowels. They can reach very high densities. You can shove a bunch of dowels into a bucket, all right? An unsaturated fat with cis bonds is like a branch. You can't fit as much wood into a bucket if it's all branches. You see what I mean? A saturated fat like butter is solid at room temperature. An unsaturated fat like olive oil is liquid at room temperature, okay? So if you've got a phospholipid bilayer that is rich in saturated fatty acid chains, then it's going to solidify at a higher temperature, okay? It's going to be easier for it to solidify, as opposed to a membrane that is rich in unsaturated fatty acids, specifically the cis bonds, okay? If it's rich in trans unsaturated fatty acids, then it's going to behave pretty similar to this, okay? But in this figure, we're talking, it tries to simplify it as saying unsaturated versus saturated. But you know from our biological macromolecules discussion, it also depends on the type of double bonds, the types of isomers, bless you. All right, so that's the saturated versus unsaturated piece. These two top things. How are we feeling about that? Same principle as with the fats. Okay. Now we talk about cholesterol. So your degree of membrane fluidity, or what level the membrane is fluid, at what level, at what temperature it becomes solid, is what I meant when I said level. At what temperature it is fluid, at what temperature range it is solid, is set by the degree of saturation in the fatty acid tails of the phospholipids, okay? Cholesterol can help to buffer that fluidity. So think about it like this. This principle sets your fluidity level, okay? And then having some cholesterol in the membrane helps to prevent the membrane from either, I'm about to wrap, so I'm not going to close the door. It helps to prevent the membrane from either solidifying, cracking, and shattering, or from wiggling apart, okay? Because these cholesterol molecules right here, embedded inside, with it, and we even have the structure of cholesterol, because it's a good figure. Okay, if you blast this with too much energy and it wants to melt and fall apart, these molecules go and then they hit cholesterol and they go boo, and they lose that energy and it gets redistributed, okay? If these things try to freeze and pack together, well, you've got this cholesterol molecule that's like, no, stop it, stop. And it's like pushing all the phospholipids apart. So cholesterol works to both maintain fluidity and prevent solidification. But it's just a buffer that maintains fluidity levels within a range set by the degree of saturation of the fatty acids making up the phospholipid bilayers. Question? It's always bad to freeze. Yeah. And whenever I say always, I'm sure there's an example of, there are examples of organisms that can tolerate this, the solidification, but it's more that they can tolerate it, not that they want it. Yeah. Awesome. Thank you, everybody. We'll do more membrane stuff on Wednesday and Friday. Yeah, have a great day. Have a great rest of your day. Enjoy the rain.